So much for Dr. Eric Chang joining us from the Feinstein Institutes for Medical Research. He's a neuroscientist who works at the intersection of electrophysiology, neurology, and immunology. He's an assistant professor at the Institute of Bioelectronic Medicine at the Feinstein Institutes for Medical Research, and he received his bachelor's degree from the University of California at Los Angeles, aka UCLA, go Bruins, and his PhD from NYU. His research focuses on how electrical and chemical signals enable communication between the nervous system and the immune system. And through the use of technologies adapted from the fields of neuroscience and bioengineering, Dr. Chang's work aims to understand how neural signals encode information about important somatosensory, interoceptive, and immune functions. His lab has recently developed tools to perform calcium imaging of vagal sensory neurons, which encode and transmit a wide range of stimuli occurring in the body. And additional work from the lab has been focused on intentionally modulating peripheral nerves, such as the vagus nerve, with optogenic or electrical stimulation to change physiological and immune responses in context of various disorders. The primary goal of his research is to determine how neural signals in different pathways encode information about physiological functions, and to use this knowledge to advance therapies in bioelectronic medicine. So welcome. Thanks so much for joining us today. I'm really excited to hear about your talk.
Great. Thank you so much, Monica. Thank you for the invitation. Can you hear me okay?
Yes, perfect.
Okay. And, I want to say, thank you to you and kudos to you and your team for organizing this series and, putting together these resources for the type 1 diabetes community. As you know, diabetes is not our primary research focus. So, just recently I was looking through some of the videos and I learned a lot from some of the different research approaches and studies that are happening in this space. So again, congrats to you for putting this together and thank you for inviting me. What I'm going to speak about today is going to be focused on the vagus nerve and particularly how it's involved in communication between the brain and the body and the nervous system and immune system. Okay. So for those of you who don't know what the vagus nerve is, it's one of the cranial nerves in the body. It's cranial nerve 10. And if you can see on this diagram on the left, it's actually a pair of nerves that descend down from the brain stem. And you can see that it is the longest cranial nerve in the body, innervating basically all of our major visceral organs. And you can see that it doesn't take a straight path to innervate these organs. It takes kind of a winding, wandering path, which is where the name vagus comes from. It it's Latin for wandering. And in some cases the innervation of an organ passes through another organ or behind it or around it. So that might present complications, which we'll maybe talk about later, about interfacing with some of these nerves as they go into end organs. It's part of the autonomic nervous system, constituting a major part of the parasympathetic pathway, which is sometimes called rest and digest, versus the sympathetic, which is fight or flight. It controls major vital functions such as breathing, heart rate, blood pressure, relevant to this audience, glucose levels, hunger, satiety, gut motility, as well as a number of autonomic reflexes such as coughing and swallowing. So you can see it's a pretty important nerve and involved in many crucial functions that are vital for our survival. What's interesting about it is actually a majority of the vagus nerve fibers are sensory in nature. So they're sensory afferents that carry information from the viscera and the body up to the brain. And we'll talk a little bit about that later on. Some of you have maybe also heard of vagus nerve stimulation, or VNS, which is simply placing a neural cuff, a stimulator, sometimes called a microregulator, onto it's simply the left vagus nerve and passing electrical current through it to activate a set of fibers that descend down into either go up into the brain or descend down into different organs. And this VNS is actually FDA approved already for a long time to treat medication refractory or treatment resistant epilepsy, then subsequently treatment resistant depression. And these are typically after patients fail several rounds. I think for depression, it's 4 different antidepressant medication regimes before they might be recommended or indicated for a VNS implant. And then just recently, 2 years ago, VNS was approved for motor recovery following stroke. And next on the horizon, maybe, is an approval for a VNS for chronic inflammatory disorders. And that came out of work that was originally actually done here at the Feinstein Institutes at Dr. Kevin Tracy's lab, where he showed in this paper a long time ago, maybe before some of you were born, in 2000, in Nature, that in a rat model of endotoxemia, showed that if you do vagus nerve stimulation electrically at the cervical level, you can reduce levels of systemic circulating cytokines in this model of endotoxemia. So if you, so I'll just go over this one figure quickly. On the y-axis, they're plotting serum levels of tumor necrosis factor, or TNF, which is a pro-inflammatory cytokine. LPS is a bacterial endotoxin that is used to induce acute endotoxemia. So no LPS, no increase in TNF. In a rat that was injected with LPS, you see this, they get sick and you see a very high rise in TNF. And then when they performed a vagotomy, the TNF rose even higher. Vagotomy is cutting the vagus nerve. And then the main result is that vagotomy, but stimulating electrically the distal ends that's still connected to the organs, actually significantly reduced TNF in this rat model of endotoxemia. So this reduction of cytokines due to electrical stimulation of the vagus nerve has been termed the inflammatory reflex, which is mediated by the vagus nerve. And work from Dr. Tracy's lab and many others around the world have shown this as efficacious, in many preclinical models. I'll just quickly go over this latest paper in PNAS that was led by a graduate student in the lab, Adam Kressel. And so what we did in this paper was activate, using optogenetics, cholinergic neurons in the dorsal motor nucleus of the brainstem to inhibit TNF. So the first thing that Adam did was he wanted to kind of clearly show the anatomical circuit, the neural circuit that descends from the brainstem dorsal motor nucleus, DMN, down to something called the celiac superior mesenteric ganglionic complex, or let's just call it celiac ganglion complex. And then ultimately to the spleen, where macrophages produce cytokines such as TNF. So what Adam did was he injected AAV, which is an adenovirus, with channel rhodopsin and YFP into the brainstem DMN. That traveled down to the celiac ganglion, and then he used a retrograde viral tracer called herpes simplex virus 1 with an M. cherry tag into the spleen. So if you look in the brainstem, this is just showing, you give it 3 to 4 weeks for the mice to express the tag and the virus. And you can see these are channel rhodopsin YFP positive neurons that are CHAT positive. So these are choline acetyltransferase positive. They're cholinergic neurons, and on the merge, you can see these are the neurons that he's later going to activate. And then when we look in the celiac ganglion, or when he looked in the celiac ganglion, I'm not going to go through all those panels, but on the first row, labeling for new N, these are neurons that are in the celiac ganglion. And then channel rhodopsin, which is coming from the AAV injection in the brainstem, co-labeled with synaptophysin, which is what SYP is. You can see an overlap. And then if we just jump to the bottom, the M. cherry neurons located in the celiac ganglion, they're M. cherry positive because they were labeled by this retrograde injection of HSV into the spleen. So if we look at O, we can see the merge of all these, which is that these are noradrenergic neurons coming from the celiac ganglion, receiving what look to be terminals from the dorsal motor nucleus of the brainstem in the celiac ganglion. And then so the functional experiments in this paper involved then performing optogenetic simulation. So optogenetics is a way to use light-sensitive opsins, they're called. In this case, we used something called channel rhodopsin 2, which is one of the most popular ones, which is activated by blue light. And you can put those the strength of optogenetics is you can put those specifically in genetically identified populations. In this case, we put them in chat cholinergic neurons. So when atoms shine blue light in the DMN, we would activate those neurons selectively. These neurons are not responsive to yellow light. So in a control experiment, shining yellow light on these neurons didn't do anything to levels of TNF here. However, when he used blue light into the dorsal motor nucleus of the brainstem, he saw a reduction in levels of TNF in a model of endotoxemia. And then he went one step further to show that if you place a nerve cuff electrode on the splenic nerve, so these were pretty challenging experiments because in a mouse, the splenic nerve is tiny. But what he did was then in a littermate control that did not have channel rhodopsin, when he turned on the blue light up in the brain stem, he didn't see any change in activity on the splenic nerve. However, when he turned on blue light in a channel rhodopsin animal, you saw a dramatic increase in levels of activity on the splenic nerve. So, this work kind of highlights and goes in this large body of literature about the descending cholinergic motor efferents that originate in the dorsal motor nucleus of the brainstem here, DMN, and then they travel down in this what are called motor efferents going down the vagus nerve to the celiac ganglion, ultimately going to the spleen and affecting macrophages that reduce levels of TNF. So, what is this circuit, the inflammatory reflex, useful for? Really for reducing inflammation in a wide variety of disorders, whether acute or chronic. And if you think about the major human diseases, just about all of them involve inflammation of some sort. So, whether it's from infection or trauma or an injury, or in the case of chronic disorders, many disorders involve inflammation of different tissue sites or the brain, neuroinflammation, including autoimmune diseases such as rheumatoid arthritis and type 1 diabetes. Monica asked me to talk a little bit about VNS and some of the work in human patients. So this is a paper that was published from Dr. Tracy's lab and collaborators in Amsterdam, as well as some people from a company that showed and published in PNAS showing that VNS, vagus nerve stimulation, can inhibit cytokine production and attenuate disease symptoms in rheumatoid arthritis patients. So what they did in this is actually an interesting, this is figure 1, I believe, of the paper. What they did was in patients that are getting the VNS implant for epilepsy, because it's FDA approved for epilepsy, they did perform some blood draws in prior to implant and then post implant, but prior to VNS. And that's what these first time points are. And then performed a human whole blood, what's called an LPS assay. And they found that in blood taken from post VNS, there was actually reduced levels of cytokines, TNF, as well as other inflammatory cytokines such as IL-6 and interleukin-1. And so that was more shown in human in vitro, but what about in RA patients? So what's plotted here is on the y-axis is a DAS-28 scale, which is disease activity score across multiple joints. So measures swelling across multiple joints in RA patients and includes CRP, C-reactive protein in the serum. So, lower values on this as we go this way are better. There's symptom improvement. So you can see on this dotted line that this is when the VNS implant took place. And this was a small study, a total of 17 patients, but you can see in cohort 1, these are actually RA patients that failed methotrexate, which is the first line indication drug treatment for RA. The second cohort, it failed multiple biologics, such as anti-TNF monoclonals. So you can see in both cohorts, the center line is them combined with the triangles, that over the course with the VNS implant turned on, their symptoms improved fairly significantly up through day 42 of the trial of the study. And then there was a 2-week period where the implant was turned off. And then you can see there was a little bit of a rebound in some of their symptoms as scored by DAS-28. And then they improved a little bit again by the end of the study. And then, so, this was in 2016. Since then, there was another human study doing a VNS in multi-drug refractory RA patients, again, that showed good efficacy for these VNS implants. So based on these human studies and then a large body of preclinical work dating back over 20 years, there's a company based out of Valencia, California called SetPoint Medical that is actually actively recruiting for a trial of RA running through the end of this year. They're currently in phase 3, and they're running through the end of this year with a target to enroll 250 patients at multiple different sites, up to 40 sites, with a double-blind sham control trial. The study is called Reset RA, and you can see this is the clinicaltrials.gov identifier. You can see what the implant looks like. It's about the size of a fish oil pill that some of us take, and has a little sleeve they call the pod that houses the microregulator. And the implant is, like I said, on the left side vagus nerve typically, and it's actually an outpatient procedure, even though it's requires a neurosurgeon. It can be done in 60 to 90 minutes, and then the stimulation parameters can be controlled wirelessly. So that's a trial that's happening now, and it's part of this emerging field that several investigators here at Feinstein are a part of, called bioelectronic medicine. And in this case, bioelectronic medicine to activate the inflammatory reflex for the purpose of treating inflammation. And so, bioelectronic medicine is this new emerging field where we want an understanding of a molecular target, let's say TNF produced from the spleen. And so, it's a combination of molecular biology to identify targets and then to understand the signals. Neuroscientists like myself understanding signals on nerves or on neurons that are involved in these circuits to mediate readouts of that molecular signal, to mediate things that are happening in an end organ or in the body. And then we work also with electrical engineers and material scientists to design and implement devices and implant devices, either in preclinical animal models or in patients, to then modify the course of disease, to treat disease. So this is a new type of therapy to treat disease with electricities and electrons, as opposed to using traditional pharmaceuticals. Let's see. So the strategy used by SetPoint is to treat rheumatoid arthritis is an example of bioelectronic medicine to activate the inflammatory reflex. There are other indications that including inflammatory bowel disease, which includes Crohn's disease and ulcerative colitis, with a similar rationale that if you put at the cervical level a stimulator and electrically stimulate the vagus nerve, that you can ultimately reduce levels of circulating cytokines, and in the case of IBD, reduce inflammation in the gut and other compartments that have an impact on patients. And sometimes people think of this as maybe competing with or replacing drugs, but in many cases, this could actually be considered an adjunctive therapy that's used in combination with drugs. So, and actually in the SetPoint trial, patients have to fail methotrexate, but while they're in the study, I believe they can still stay on methotrexate. So again, the end goal is to improve symptoms and ideally cure disease and disorders. So if it requires a combination of a nerve cuff electrode and implant in combination with the pharmaceutical regime, and that's what's needed to improve the patient's symptoms, then that's what we should go for, right? So up to this point, we've focused on the descending motor efferents that are in this inflammatory reflex mostly, but if you recall from the beginning, what I said is that the vagus nerve is actually 90%, up to 90% sensory afferent. So if we look at the other side of the coin or of the nerve, we can see that the sensory information coming from the body and different tissue compartments going up to the brain can travel either through dorsal root ganglia and the spinal cord through the spinal chain, or the other part which I'm going to talk about now is the vagus nerve also sends visceral, sometimes called interoceptive information up to the brain via the nodose ganglion, which you're going to hear about next. And then the next synapse after the nodose, so that's the last kind of part of the peripheral nervous system before we go into the CNS, is the NTS, is the nucleus tractus solitarius, part of the brain stem, and then that sends information to other parts of the brain. So if we look at just the sensory vagus nerve, one of the big questions we're working on in the lab now is how is inflammation and other types of stimuli that are happening in the body sensed by the peripheral nervous system, sensed and encoded by the peripheral nervous system. So you can see, again, that the vagus nerve innervates all these organs in our body. I highlighted the pancreas here for this audience. So what we want to know is when there's injury or inflammation happening in a part of the body, there's a typical inflammatory response, and critical to that is a cytokine release, such as tumor necrosis factor, which I already spoke about, IL-1, IL-6, and sometimes IL-10 anti-inflammatory cytokines as well. So we want to know how are those things encoded by the vagus nerve. And so, like I said, when there's inflammation in different organ sites, how does the brain ultimately know about that, and how is that encoded by, we think, by these nodose ganglion neurons, because these are where the cell bodies of the sensory afferents reside. So in that PNAS paper with optogenetics, we activated the cell bodies of the efferents that go down to the organs, those are of the cholinergic efferents, but the cell bodies of the sensory afferents that go up to the brain reside here in the nodose. So this is a paper that our lab published just last month. And this was work led by Tomas Huerta, who's currently a postdoc in the lab. And what we did here was develop a set of tools and techniques for imaging this nodose ganglion, which houses these vagal sensory neurons. And you can see that where the nodose ganglion is just kind of this little bump here on the vagus nerve. And because of where it's located at the base of the skull, and this is also the case in humans, is at the base of the skull, at really high up in the neck, it's in an area that's very difficult to access. So if we wanted to image those nodose neurons, Tomas and the lab developed a technique, surgical approach that basically, I'm not going to show you the surgery images, but under anesthesia, you can open up the neck of the mouse and then find the vagus nerve, see as it crosses another cranial nerve, hypoglossal. And then this, at the bottom of this is as we go down closer to the skull, that's where the nodose ganglion is. And then we developed a set of tools, a set of approaches to stabilize the nodose. And then while it's still intact, fully intact and connected to the vagus nerve, so it's still connected to all the organs of the body, we use something called a miniature microscope or a miniscope to then image the nodose intact and then apply different agonists that I'll show you next to the vagus nerve just below it. And we did this in mice that have a genetically encoded calcium indicator called GCAM6F. And we did this in mice that have only this calcium indicator in glutamatergic neurons. So these are in VGLUT2 GCAM6F mice. And we did that because we know that these vagal sensory afferents are actually glutamatergic. The vast majority of them are glutamatergic. So in these mice and in these nodose, when there's neuronal activity, we see changes in fluorescence that we'll be able to image with the miniscope. And then we develop a set of tools on the software side on some Python-based software packages that allow us to take the identified neurons where they are in space in the nodose and then plot what are called calcium traces, which are basically changes in fluorescence over time. So this is what it looks like when we actually image the nodose. And in this case, this is to an application of capsaicin, which activates TRPV1 channels, which many of these nodose neurons have. And you can see this is kind of like a raw video, raw data, which is essentially videos. And every time you see, basically these white spots flashing, that's a change in calcium fluorescence. That is an indication of these neurons actively firing. So this is a figure from the paper that was just published. And again, you can see more details about it if you're interested in the paper. But we developed tools to identify the regions of interest with the neurons, then plot the traces to specific chemical stimuli, in this case, capsaicin, and then select out and filter out the neurons. Some of these neurons respond to different things like mechanosensation or breathing or things that are happening with different organs, because this nerve is connected to almost all the major organs. So we want to know just the ones that respond to our chemical agonist. And then in addition to capsaicin, we decided to, because we're interested in inflammation, to look at these other channels, TRP channels. So capsaicin, as I said, is activating TRPV1, but transient receptor potential channels such as TRPV1 and TRPA1, which is ankyrin 1, is another channel that is located on these nodose neurons that have been linked with inflammation. TRPA1 is activated by thermal changes such as noxious cold, cinnamaldehyde, mustard oil, as well as a chemical called AITC, which is what we ended up using. TRPV1, many of you may have heard of, is what's activated by the capsaicin, which is what makes chili peppers hot. It's also obviously then also activated by changes in heat and acidity. And then we know also metabotropic glutamate receptors are on these neurons. So another agonist we use is glutamate. So using these different classes of agonists on the vagus nerve, we then wanted to look at different components of these calcium transients. So we quantify the peak of the responses, the number of peaks, the duration, and the integral. And what we saw is that when we applied capsaicin or glutamate or AITC onto the vagus nerve, and you can extrapolate out one step and say, what if capsaicin was in the gut or in the pancreas or in the liver, that there were certain signatures that were unique to each class of agonists. So for example, capsaicin had a higher peak amplitude, but a lower duration when you compare it to glutamate. So these seem to be certain, characteristic signatures of each class of chemical agonists. And then what we did next was then we take all these traces and we wanted to see, well, if we just looked at the neural activity in the nodose ganglion of these vagal sensory neurons, could we tell that which agonist was applied to the vagus nerve without knowing that? So what we then do is use a machine learning random forest classifier to put in several of those components. We chose 4 of them: amplitude, duration, number of peaks, and integral, into a random forest classifier, and then basically it, based on those features, would be able to classify this trace was, capsaicin was applied, or this was AITC was applied. And so the result, this is a figure in the paper. This is the receiver operating curve, the ROC curve for the performance of the random forest classifier. And you can see for capsaicin, for example, the model was had an accuracy of about 82% for just looking at the traces and classifying that as capsaicin. It was 78% for glutamate and a little lower, 71% for AITC. Another thing that we found was interesting is that if you look at the Shapley explanatory values, or the SHAP values, that there are certain features of those calcium transients, particularly the duration and amplitude, that had a particularly high impact on the performance of the random forest model. So, what this tells us is that the information that the vagus nerve is seeing, as far as specific chemical agonists, in this case capsaicin, glutamate, and AITC, activating different channels, is encoded in a particular way by these neurons in the peripheral nervous system before it even gets to the brain. And what we're really interested in, and this is work that is in progress that we're working on now, and again led by Tomas Huerta, is that we want to know, are there cytokine specific responses? So when we apply IL-1 to the nerve, or when IL-1 goes up in the lung or in the liver. And you can see in this video that there's a single neuron here that seems to be robustly responding to IL-1 in a characteristic fashion, kind of in a bursty fashion. What are the cytokine-specific neural responses? And that gives us insight into how the peripheral nervous system encodes aspects of inflammation. As I said, we're interested. Also, we have data recording of when TNF is applied. So you can kind of see if you compare and contrast the capsaicin video that the cytokine responses look pretty different. And as I said, we're working on this now, and what we're starting to see in a similar way is that there seem to be characteristic patterns for IL-1 neuronal responses that are different from those to TNF. We also have data I'm not showing here on IL-10, and this starts to put together some rules for how these things are encoded by these neurons. And we also have data in a disease model of chronic inflammation that, so stay tuned. We're working on that paper now. But basically, there seem to be very significant differences that happen in the nodose ganglion when there's a chronic state of elevated inflammation. Specifically that these neurons and vagus nerve activity seem to be highly increased in those states. So just to then bring some of this work back to the vagus nerve and type 1 diabetes, which this is what this audience I think is interested in. This is work that maybe some of you are familiar with from a lab at University of Miami, Alejandro Casero's lab, so this is not our work, but you can see the vagus nerve also innervates the pancreas, and you can see this very nice images they have here, substance P to label the sensory aspects that go into the pancreas, and you can see that the innervation is very close to the islet cells. And what they showed in this paper is that the beta cells in the pancreas release insulin, but they also simultaneously release a neurotransmitter called serotonin, or 5-HT. So that work like this and others are starting to reveal at a molecular level and at a neural level some mechanisms of communication between different pancreatic cells and the vagus nerve. We're starting to understand how that happens, and this has implications for signaling to different areas of the brain. And throughout all this, I haven't talked about the brain at all, so I hope the neuroscience gods will forgive me. But the brain obviously has important roles and functions as far as providing, particularly, let's say, the hypothalamus for top-down control of insulin and potentially blood glucose as well. VNS, vagus nerve stimulation, and other bioelectronic medicine approaches, such as stimulation of other nerves that innervate end organs like the pancreatic nerve, are and have been explored for the regulation of blood glucose in hyperglycemia. And of course, the regulation of blood glucose, something as important as blood glucose, is likely to be multifactorial with multiple redundant molecular and neural circuits that are involved in its regulation. So, people want to think that the vagus nerve does everything and can control everything, and it's involved in many things. But there are obviously redundant mechanisms in place, including the brain. Let's not forget about the brain. So from the clinical perspective, some symptoms of type 1 diabetes have been associated with damage to the vagus nerve, including gastroparesis, a slowing of gastric motility, which are associated with heartburn, nausea, and abdominal bloating. And there seem to be a handful of papers showing that there's actually vagus nerve atrophy in some diabetic patient populations. So, there is evidence of a clear connection, and then work from labs such as the one work from like this on the left, showing increased understanding of what exactly the vagus nerve is doing in its innervation of the pancreas, and then of course how is the brain involved in some of this regulation as well. Our lab at the Feinstein has published a little bit of work in this space, and this is a work done with Dr. Tracy's lab and another lab here, Dr. Theo Zanos, who's works in artificial intelligence and machine learning and data analytics. And this was work led by Emily Bandinelli-Massey, who was another graduate student in the lab. And what Emily did was perform vagus nerve recordings. This is under anesthesia, but basically you have a nerve cuff electrode interfacing with the vagus nerve, hook it up to an amplifier, and record vagus nerve activity, of which there's a lot of activity, and it's very noisy because of all these signals going up and down to the brain and the body, between the brain and the body. But what Theo's lab did was use some data analytic tools to extract basically what are called compound action potential clusters, CAP clusters, from this very rich signal. And what they found was that if you're doing Emily performed the vagus nerve recording, and then at 30 minutes in, you're tracking these clusters as they come in and out, and you saw that following an injection IP of insulin compared to saline, that there were certain groups, CAP clusters, waveform clusters. These aren't necessarily individual fibers, but they're potentially groups of fibers and groups of neurons that became active following insulin injection. So it appears from this work and a handful of others that the vagus nerve does carry information about insulin specifically, and whether or not these CAP clusters and these fibers or neurons are related to these vagal sensory axons that they worked on in this paper. It remains to be seen, but that's a possibility. Of course, this audience is probably aware of artificial pancreas, which is a device implant to release insulin into the body. Of course, for bioelectronic medicine, what we try to aim for is a closed loop approach, which is having a sensor, whether that be a nerve cuff or another sensor in a different part of the body that can sense changes and signal the device to release insulin. There's the next generation of this artificial pancreas, which can release insulin and glucagon. So this technology is only going to improve. I think in this space, what we're missing is a good understanding of the neural signals that are important to implement a real closed loop system that would really be beneficial over standard treatment for patients. So let's see, this is my third to last slide, I think. But to sum up the many different functions and talents of the sensory vagus nerve, now you've seen many different schematics and versions of this, but basically we know that the sensory vagus nerve innervates all these organs. And I didn't have time to talk about it today, but our lab has also published work activating with focused ultrasound stimulation. This is a collaboration with GE Research, stimulating the sensory innervation of the liver at the porta hepatis. And if you're interested in that, that was Tomas Huerta was the lead author. That was published in 2021. And I'm not going to go through all these, but basically, we've known for a long time that the vagus nerve is involved in many of physiological functions and innervates many of these end organs, almost all the major end organs. But now, and this is why so many labs and research groups are potentially interested in it, including ours. But the vagus nerve has clear involvement in many different functions and potentially disorders. So your disorder of interest that your lab studies, you can probably find some vagus nerve involvement if it's innervated by the vagus nerve. But what's starting to change in the last 5 to 10 years is that we're really starting to see, we as a community are starting to see what are the actual receptors and molecular mechanisms that are involved in some of these reflexes that have been known for 100 years or more. That lung stretch reflexes, stomach distension. We know that the vagus nerve is involved in these, but now using modern tools that include optogenetics, molecular genetics, Cree-based mouse lines, and improved technology, we're starting to really understand that piezos, those channels are involved in this exact reflex and this exact function. Showing that serotonin is involved in signaling from the pancreas up to the brain is another example of how we're getting a better handle on how does the vagus nerve perform all these different varied functions. So just to summarize, this is a very famous illustration from the neuroanatomist Andreas Vesalius. You can see when it was made. So, 500 years later now, we've known for a long time the macroscopic organization and the fact that this complex nerve innervated all these tissue compartments and organs. But what's changed really in the last 10 years or 5 years is that there's advances in genetics such as single-cell RNA-seq, advances in indicators like in neuroscience and technology such as the GCAM6 we use. There's also voltage indicators that are being developed, and as well as technologies to image them at a high resolution, both spatial and temporal. Using these modern tools, we can start to reveal some of the coding strategies used by the vagus nerve to transmit all these signals up to the brain and allow us to survive. Our lab has, very recently with this last paper, contributed a small part to this by developing a set of tools to perform in vivo imaging of these vagal sensory neurons as they respond to different specific chemical stimuli and inflammatory mediators. And we're working now to see what's the coding pattern that these neurons use for cytokines. And what we know already is that these signals are encoded in the periphery before the information is sent to the brain. And new approaches I think are going to come out in this field of bioelectronic medicine that will hopefully be able to capitalize on some of this understanding of these neural signals on the vagus nerve and of these neurons to treat different inflammatory conditions. The company is already, SetPoint is already in phase 3 trials for RA, so that's one that's on the horizon. But other autoimmune disorders are certainly possibilities, as long as we understand the molecular basis of what's going on in the neural circuit that might be manipulated to treat those diseases. So finally, I want to thank you, everyone who's online, for your time. Thank you to Monica and Sugar Science for the invitation. And I want to thank, I work closely with several faculty members here at the Feinstein Institutes. And also, this is a subset of our larger lab group, including specifically the people that contributed to the work that I showed you today. I want to thank and acknowledge our funding sources. And if anyone has any questions for me afterwards, here's my email address and my Twitter handle. And thank you, and happy to take any questions.
Thank you, Eric. That was excellent. It was really, it's fascinating how you and your team are really kind of going after understanding the crosstalk between organ and brain. I had a couple questions, and anyone is free to, raise your hand. So I had a quick question. So in many autoimmune diseases, not only do the sympathetic nerves become overactive, they kind of rearrange themselves in a pro-inflammatory circuit, and then in response, the vagus nerve, which opposes them, becomes underactive, right? And so, do you know of anyone who's done the work, sort of like, recording the vagal nerve in those who are at risk for autoimmune diseases like type 1 diabetes before and after their diagnosis?
No, I'm not aware. So obviously recording from the vagus nerve is very challenging.
I'm sorry, not recording, but sort of like maybe even looking at like reduced RHV, heart rate.
Yeah. So right. So the standard autonomic measure being used is heart rate variability, which is a kind of a proxy for vagal tone. But at least in my estimation of it, it seems to be a very unreliable. It's not really a robust measure. And really what some groups, including some labs here at Feinstein, are trying to do are to actually perform recordings of activity like what I showed you. That study was done under anesthesia. And just because of where the vagus nerve is in the neck, there's a lot of movement artifacts when you try to perform chronic recordings. Our lab has worked with other labs here to develop and publish techniques on doing chronic recordings. And I think that's happening now, and I think we'll hear more about what exactly different parts of, just the cervical level, you kind of catch all the information that's going up to the brain and also going down to organs. We really need to get closer to the end organs and try to record and stimulate at those sites. So to come back to your question, I'm not aware of people with even HRV readouts before and after diabetes, although there may be some. I think what's clear is that there is autonomic involvement in a lot of these disorders, and it's becoming bigger following COVID too, right? So, I think the non-invasive measures, is particularly in humans, are challenging. There's a lot of noise, and I would not say they're robust. Short of performing an actual cuff recording on a human nerve, which actually some groups have done. I can send you those papers later. I don't recall the groups, but it was just last year, a couple groups have actually done human vagus recordings.
That'd be great. We'll post those.
Yeah, the HRV is not particularly reliable. It's just what people have at hand and what you can have as a non-invasive readout.
Right. And just sort of that, to follow up on the sort of this tracking people in this upcoming set point RA trial, will T1D as a comorbid condition be tracked at all or captured?
That I don't know. I mean, we don't have any involvement with the company. I don't know if there's any type 1 diabetes component to that at all. But I can find out for you.
It would be so interesting to capture that cohort.
Yeah, I mean, you can see on clinicaltrials.gov what the eligibility criteria are. So it may be even that if you have other comorbidities that you might be excluded from that trial, because they probably want it to be as clean as possible. But I don't know if that's. I know that they're looking at other things like multiple sclerosis, for example, and other things, and there are many other groups looking in type 1 diabetes for sure.
Oh, great. The paper of the VN recordings in humans Dr. Chang was referring to is now been posted in the chat by Dr. Amparo Guermes.
Great, thank you.
Hi, yes, sure. Thank you so much for the super interesting talk. So I also work on recording from the vagus nerve, and so I'm quite into the field as well. And one of the questions that people keep asking me is that, because I envision also like this closed loop control, adding the neuromodulation as well. And people constantly ask me how is it feasible to do an invasive approach? Because of course, like for recording, as you were saying, like non-invasive strategies are really not reliable enough. So like people with diabetes that they manage to control their disease in a non-invasive or minimal invasive way, whether this is something that makes sense. And I would like to get your opinion on that, because I mean, I have mine, but I'm always happy to get other feedback.
Sure. Yeah, I mean, it's certainly very challenging. That doesn't mean that it's impossible. And we and other labs here at the Feinstein, as well as other collaborators in the US, have developed techniques to do chronic recording of the mouse cervical vagus, which is a 80 to 90 micron diameter nerve that's constantly moving around because of where it is. I think I'm hopeful that bioengineers and the people like in your group as well that you work with will be able to develop techniques maybe at the cervical level, because there's so much information going up and down and it's a mixed fascicle. We do a lot of work in mouse and rodents, but obviously other groups work in sheep and pigs and humans, which the fascicular organization is different. And there are different strategies that other labs use to address certain fascicles in the cervical level. But in some cases, it might make more sense to do an implant or a stimulation or a recording device near the end organs, right? Yeah. So, as far as, the bioengineering aspect, I think, and there are other groups like, I believe John Rogers' group, and Tim Brunn's group at Michigan have recordings of nerves that go into the bladder for bladder control, so looking at the signals that are going close to an end organ might be more useful if you're trying to treat a disorder that organ's the primary target. rather than going up to the cervical level, which has different challenges. And obviously, even if it's like a neural interface at the pancreas or at the bladder, obviously there's still some movement, but it might be less than in a highly dynamic region like the neck.
Yeah, totally.
So I think and like you shared this paper, I mean, it's possible to do it in humans.
I was so shocked when I read it. Like they basically put a
Yes.
tungsten microwire inside the vagus. So I was like, wow, I'm
Yes.
struggling. Like I'm just working with rats here. Like of
Just
course, like
rats.
I dream of pigs, but like humans, it's it's a different level.
Yeah, and yeah, so obviously the pig fascicular organization is closer to the human. It's actually more fascicles than a human. But then the stimulation and recording strategies for these different types of organization will have to differ. So the electrode design will have to differ. And Stavros Zanos's lab here at the Feinstein Institute has a grant to study exactly that question and to map out the organization in the human vagus nerve from human cadavers.
That is very interesting work. Well, thank you so much.
Thank you.
Well, yeah, Dr. Amparo Guemes coming from Cambridge University, and sounds like maybe there's a conversation that could be continued to foster some collaboration.
Absolutely, yeah.
Fantastic.
And thank you, Monica, for hosting this once again.
Oh, no, this is a very interesting field, and it's growing so quickly, and I think there's a lot of runway for inquiry here in terms of type 1 diabetes. So we're really following young I would say up and comers and contributors like yourself and Eric Chang. I wondered, I see that Kevin Tracy is here. I wondered if he might unmute and just sort of share his vision for the ultimate, application of some of the technology that the group is developing in terms of autoimmunity.
Well, sure. Hi.
Hi.
First, I want to congratulate Eric for his talk. It was great. I learned a lot, actually. I'm going to have Eric give the same talk now to the lab, many of whom are not on this call. But I think that there's opportunities through recording or visualizing, as Eric's doing, or through deriving additional maps to just think differently, first of all, than what the textbooks say. Eric really, I don't think he did in his talk, and usually doesn't, I never do, talk about sympathetic versus parasympathetic, because by the movies that you watched, the function is much more complicated than left or right, sympathetic or parasympathetic. I think we have the tools in place now that, as Eric was talking about, to ask questions about individual fibers, and not what family they belong to, but what functions do they control at the cellular or mechanistic or molecular level, and how can we manipulate them in the context of a therapeutic question or goal? And I think that's the overall, that's the future of this field, bringing together neuroscience, endocrinology or immunology, physiology, call it, and biomedical engineering. It's going to change how people think about designing therapeutics, because if you have a molecular target in mind, then your challenge is to map the sensory fibers to that target, and or the motor fibers to that target, which can affect a change in the tissue. So yeah, I think that's the vision. I think the vision is that if you can map a disease to a specific molecular mechanism, then it is now possible to at least frame the question: are there neural circuits that reflexively target that mechanism? And if so, can they be hacked to develop new therapies?
Yeah, that's perfectly put, right? First the cartography, then the hacking.
Yes.
Yes, agreed.
That's great. Just, hats off to all you're doing there at Feinstein and Northwell, and, around the world, around the globe, as people address bioelectronic medicine in context of autoimmunity. Thank you both again, and everyone who attended. And we look forward to the next papers coming out of the laboratory.
Great, we'll let you know.
All right, we'll be
All right.
watching. Have
Thank
a
you so
great
much,
rest of your
Monica.
day. Thanks again.
Okay,
Bye-bye.
you too.